We are starting a new chapter in the unit of plant physiology, that is transport in plant. Transport in plants. Now we need to know what all things are to be transported in plants. There are three categories or three substances mainly. One is water, which needs to be transported. Then there are minerals which need to be transported. So water and minerals, and third thing is the food which is synthesized by the leaves. So food has to be transported from leaves to various storage organs or wherever they are required by the plant. So these are the three things which are to be transported. Now if we talk of how much distance do these things need to be transported, that can be studied as short distance transport if they are to be moved only to small distances, that is from one place to next place which is close by or within a cell. So short distance transport and long distance transport. Short distance transport normally takes place by diffusion and by cytoplasmic streaming. That means cytoplasmic streaming, that means by movement of cytoplasm. So if it is to be short distance, then diffusion is the most common process, or if it is within the cell, then just circulatory movement of cytoplasm that would help in transport. So short distance transport of substances by these two methods. Long distance transport is normally with the help of vascular tissue, that is xylem and phloem, and this movement of substances to a longer or greater distance which is done by vascular tissue is known as translocation, which is normally done for the water which is taken from roots, root hair, and then it has to be transported to the topmost part of the plant. Through phloem, the food which is synthesized by leaves and to be transported to the storage organ, and if that storage organ is in the root, then from topmost leaves to that storage organ. So this is long distance, and short distance can be only within the cell or between the cells. So these are the processes. Now let us talk of the various modes by which this transport can take place. That means if we are talking of this transport, then what are the means of transport? When we talk of means, we talk of two processes again, a passive process and an active process. Passive process is without expenditure of energy. So there is no ATP consumption. Whereas active transport is always with the use of ATP. So ATP is required, or we can say it is a must. In absence of ATP, active transport will not take place. Second important difference between these two means is, in case of passive, it can only take place from higher concentration level to lower concentration level. So from higher to lower concentration only, and this can take place against the concentration gradient. So can take place even against concentration gradient. This means that if there are 100 molecules on one side of a membrane, then only 50% would get transported. Suppose this is the cell that we are talking of. There are 100 molecules outside, and if we are talking of passive movement, then a stage will come where inner compartment will have 50 and outer will have 50. We call this condition as equilibrium. And as there is no concentration difference, no net movement of molecules would take place. But if it is taking place with expenditure of ATP, then all 100 molecules can be taken in. That means there would be a stage that inner would be 51, outer would be 49, or even more. Inner would be 80, outer would be 20. So this is taking place even against concentration gradient because energy is consumed. Another important difference between these two processes is that passive transport or passive movement is not that sensitive to temperature and inhibitors. So, not sensitive to factors like temperature and inhibitors. Whereas active transport is very sensitive to these things. So it is very sensitive to temperature as well as inhibitors. Because to use this ATP, there are enzymes and there are proteins which are required. And if there are proteins and enzymes, we know protein gets denatured in case of higher temperature, as well as inhibitors are going to reduce or affect the activity of enzymes. So active process, ATP dependent, against concentration gradient also it can take place, and it is sensitive to chemicals which come in the category of inhibitors, and if the temperature is higher, then also they would get affected. Now let us talk about the various processes like diffusion, facilitated diffusion and osmosis, and all these three are passive processes. So let us come to various types of passive processes now. Let us talk about the process of diffusion. Diffusion is defined as transport of solute particles from the region of higher concentration to the region of lower concentration till it reaches equilibrium. So it is number one, movement of solute particle, and solute particles can be gaseous, liquid or in solid state. So this is point 1. 2, it is always from higher concentration of the solute to lower concentration, or the region where the concentration is lower, from this region to that region, and it is still equilibrium. And we already discussed that this is a passive process. Passive means it is without expenditure of energy. So certain particles, solute particles, move from the region of their higher concentration to the region of lower concentration till this reaches an equilibrium. That means 50-50 distribution of the particles without expenditure of energy. That means it is a passive process. Now, in case of diffusion, there are certain factors which affect the rate of diffusion. So let us talk of factors affecting the rate of diffusion. The first factor is temperature. Higher the temperature, faster is the rate of diffusion. In junior classes, we study that hot food or the food when it is being cooked can be smelled from a greater distance as compared to the food which is kept in a refrigerator. The reason is when you're cooking it or when we are heating it, the kinetic energy of those particles is more because you're providing heat. So heat energy is getting converted into kinetic energy, and these particles, they are able to travel a greater distance. So temperature factor, higher temperature, rate of diffusion is faster. So faster diffusion. Second factor is density of the substance which is getting diffused. So when we talk of density of the substance, we need to talk of a law which was given by Graham. So it is known as Graham's law, which says that the rate of diffusion is inversely proportional to root of density of that particle. That means if the particle which is diffusing is heavier or denser, it will not be able to cover greater distance or longer distance. 3. Concentration gradient. Concentration gradient. If the concentration gradient is more, the rate of diffusion is going to be faster. What exactly we mean by this is, if one side of a compartment or a room has very high concentration of the solute and the other corner or other compartment is not having any of that particle, then the movement is going to be faster. Say we talk of this particular thing, and say in this tank of water we just put few crystals of potassium permanganate. So this concentration is maximum in this corner and there is no big particle in any other region. So diffusion is going to be faster from the highest concentration to the area where concentration is absolutely zero or negligible. So higher the concentration gradient, faster is going to be the diffusion. 4th, surface area. Surface area. If area through which diffusion is taking place is more, then the rate of the diffusion is going to be faster. We can understand it from the example of our lungs. In our lungs, the alveoli, they make about 100 square meters of surface area, which is a very good respiratory surface for exchange of respiratory gases, because this area is greater. So if greater surface area, rate of diffusion is going to be faster. Another important point which we need to know about diffusion is that if the particles are lipid soluble, they'll diffuse faster. That means lipid soluble particles move or diffuse faster, because the diffusion is taking place through the plasma membrane, and if they can dissolve into or they can move through this lipid layer of the plasma membrane, then they would be able to diffuse faster. So these are the factors which regulate the rate of diffusion. And whenever we're talking of diffusion, it can be gas in gas, liquid in liquid, gas in liquid, solid in liquid. Now diffusion of solid in liquid or gas takes place when we spray a perfume or when there is an odor, then these are solid particles which are diffusing into the air. But diffusion in solid is a very, very slow process, or in certain solids it does not even take place. So diffusion of solid is possible, but diffusion in solids is not possible, or if at all it takes place, it is a very, very slow process because that solid is a very dense medium. Let us talk about the next process by which this transport or exchange of substances takes place. The process is known as facilitated diffusion. Now here there are two terms which we need to understand. The first one is facilitated and diffusion. We have already discussed about diffusion. So here it is passive movement of particles, but it is helped or facilitated by carrier proteins. So there are carrier proteins which are going to help in transport of these substances. Now how do these proteins work? Suppose we talk of the structure of plasma membrane. We know that here there are phospholipid molecules and there are extrinsic proteins, intrinsic proteins. So if we just draw a couple of intrinsic and extrinsic proteins, this is outside and this is the inner side of the membrane. Now if this protein particle is acting, or protein molecule is acting as a carrier, it is going to bind with that molecule which needs to be carried in. This is our molecule which has to be transported. So step number 1, the molecule which is to be transported binds with this carrier protein. and then this carrier protein, it moves into the membrane. So it comes here, the next stage will be here, and every time when it is moving, it is coming with this carrier molecule. When it comes on the inner side, it releases that substance which was to be transported, and this protein is going to go back. So the protein moves, but the interesting part is that there is no energy which is required. There are two important proteins which we talk of when we talk of facilitated diffusion. One are called porins, which make large pores in the membrane. And these porins, they are normally found on the outer membrane of mitochondria and plastids. And these porins, they are also found on the membrane of some bacteria. These are one protein, and the second proteins that we are talking of which help in this facilitated diffusion are called aquaporins. Aquaporins. And as the name says, aquaporins, they are helping in the transport of water. So they are also known as water channels. There are two types of particles which are transported with the help of this process, that is facilitated diffusion. These particles can be polar or non-polar. Polar particles, if we have to take examples, these could be sodium ions, chloride ions, and if we have to talk about non-polar molecules, then nucleosides, amino acids, these molecules can be considered as the non-polar molecules which are taken in by facilitated diffusion. Important thing is, it is a passive process, there is no energy which is required, and there is a protein which is known as the carrier protein. There are many carrier proteins which work for this, and these proteins, they bind with this substance which is to be transported, move through the membrane, come on the inner side, release that substance on the inner side, and then go back. But doing all this, no ATP or energy consumed. So it is a passive process, but the transport is facilitated by some special type of proteins. And these two proteins, that is porins and aquaporins, are a few important ones. Both the type of molecules which get transported, polar as well as non-polar. So these are passive methods. One more passive method which takes place is osmosis. But before we talk of osmosis, let us first talk about the direction ways. If we can classify this like Uniport, Symport and Antiport transport across the membrane, because here also there are proteins which are going to help in this transport. So let us talk of these three first.